The International Space Station might be a technological marvel, but it doesn't offer the comforts of home. Because this is a concentrated universe where living is anything but easy. Space is limited, putting great pressure on the astronaut's body and mind. Muscles waste and the atmosphere is claustrophobic. When manned deep space missions become a reality, it's going to get worse. Scientists are looking to the animal kingdom for answers, specifically tracking down the mechanism that allows them to hibernate, an ideal condition for astronauts traveling for years through space. At the University of Pavia in Italy, a research team is conducting hibernation experiments on behalf of the European Space Agency. The team have been able to create a hibernation-like state in cell cultures using a synthetic molecule called daddle, which has opium-like properties.
Vedere se per mezzo di questa molecola che si chiama daddle.
Daddle is similar to a family of molecules produced in the brain. It reduces energy consumption in cell cultures, but also in animal cells and the cells of other organisms.
In a nutshell, the team have found how this molecule reduces the energy consumed by the cell and basically puts it to sleep. But this may only be the beginning.
We can say that we are very happy to have understood the basic mechanism, to see how the molecule affects the cells, and to try and produce a state of extremely lowered metabolism with animals. And why not, one day, do the same with man?
Researchers at the University of Verona, some 200 kilometers away, have been testing the daddle molecule on non-hibernating animals, starting with rats. The rodents injected with daddle and its subsequent behavior compared to that of a non-treated animal. Sensors track its heartbeat and brain activity. Because hibernation in animals is characterized by huge drops in heart rate, body temperature and metabolism. Finally, the rats are anesthetized and studied using a magnetic resonance scanner to check the state of its organs.
The preliminary results we've obtained show essentially a certain reduction in body temperature during the 4 hours after the daddle was injected. This lowering of temperature is associated with a net diminishing of movement.
Injecting human hearts or lungs with a hibernation-inducing agent could preserve them for transplants, or a severely wounded person could be put in hibernation until treatment's available. But such miracles are a long way off.
We're trying to learn more from naturally hibernating animals what the mechanisms at work are. One day all will be understood. Then it will be much easier using pharmaceutical techniques or other methods to apply these mechanisms to non-hibernating animals.
The discovery would pave the way for the human hibernation of a kind foreseen in the film 2001: A Space Odyssey. Crews could be put to sleep for months or years, meaning less space and food. The spacecraft would be lighter and easier to maneuver. Its detractors say human hibernation is sheer folly, but its supporters counter that long ago it's believed man's forebears used to sleep through the long cold winters. So perhaps astronauts of the future will be discovering human secrets of the past.